Hello learner, welcome to the first lesson in the first topic of Form 2, which is transport in both plants and animals. And in our introduction, we need first to know what transport is. Transport is defined as the movement of substances from one part of the body of an organism to another. It involves movement of materials from the environment to the sites where they are required, and then removal of metabolic wastes from their sites of production to their sites of elimination. Remember, nutrients and oxygen are the raw materials needed by the cell, and they must be transported to all living cells of the body for various metabolic processes. These metabolic processes in the cells produce excretory or waste products like carbon 4 oxide and nitrogenous wastes, which should be eliminated before they accumulate or collect in excess in the cells. Otherwise, if they are left to accumulate, they become poisonous or toxic to their cells. That means accumulation of these metabolic wastes will kill the cells. That is why excretory products should be transported to their sites or parts of excretion or removal. Organisms like amoeba are unicellular, meaning they are made up of only one cell. They have a large surface area to volume ratio, and that is why their body is in contact with the environment. That means the process of diffusion is adequate to transport substances across the cell membrane and within the organism. And remember, diffusion is that process whereby particles or molecules move from a region of high concentration to the region of low concentration across a semi-permeable membrane. And the cell membrane is semi-permeable. That means it allows only substances that are small in size to pass through, but not large ones. Large multicellular organisms have complex structures where cells are far from each other. Therefore, diffusion alone cannot meet the demand for supply and removal of substances. That means an elaborate transport system is important for supply of materials such as nutrients and oxygen. At the same time, rapid removal of metabolic wastes like carbon 4 oxide and nitrogenous wastes like urea. In plants, certain metabolic wastes are converted to less toxic forms and transported to certain plant organs for storage. That is why plants do not have complex excretory system. And even plants can reuse some of their wastes. Simple plants like mosses and liverworts lack specialized transport system. Transport of substances takes place through osmosis, diffusion, and active transport. So refer to Form 1 so that you can remember these processes. Higher plants have specialized transport systems known as the vascular system, which has the phloem and the xylem. Xylem conducts or transports water and mineral salts. On the other hand, phloem translocates or transports dissolved food substances like sugars from the leaves where they are manufactured to the rest parts of the plant. To understand the mechanisms involved of transport in higher plants, it is necessary to study the anatomy or the structure of root and stems in relation to transport. We shall look at the internal structure of root and root hairs first. What are some of the functions of the root? One is anchorage, that is holding the plant strongly to the soil. Absorption, that is uptake of water and mineral ions from the soil. Roots also perform storage, as seen in sweet potato plants. Gaseous exchange, as seen in mangrove plants. This is an internal structure of a root, a dicot root. You can see these are the root hairs. We have the polyphorous layer, we have the cortex, then we have the phloem, and this one, which is the star shaped, is the xylem. This is a monocot root. You can see it has a space at the center called the pith. And then we have the endodermis, we have the cortex, root hair, and the polyphorous layer. So this is a monocot root. This is a transverse section of a dicot root showing the region of maturation, then, or specialization. We have the region of elongation, the cells elongate, and then here we have the region of cell division, and then the part that is responsible for this rapid cell division is the apical meristem, and it is covered by a root cap. So the outermost layer in a root is the piliferous layer, and this is the special epidermis of young roots, whose cells is to give rise to root hairs. And root hairs are microscopic or small or tiny outgrowth of epidermal cells. They are found behind the root tip. They are one cell thick for efficient absorption of substances. They are also numerous or many in number and elongated to provide a large surface area for absorption of water and mineral salts. Another adaptation is that root hairs penetrate the soil and make close contact with it. Just below the piliferous layer is the cortex and contains loosely packed thin-walled parenchyma cells. Water molecules pass through this tissue to reach the vascular bundle. The cortex acts as a storage tissue because of these parenchyma cells. In some young plants or young stems, cortex cells contain chloroplast. The endodermis, or also called the starch sheath, is a single layer of cells containing starch grains. This endodermis has what we call Casparian strip, which has an impervious deposit controlling the entry of water and mineral salts into the xylem. So this is a root hair cell. It has a cell wall, we have a nucleus, then the cytoplasm. The whole of this space is the vacuole. Just below the cell membrane or the cell wall is the cell membrane. Pericycle forms a layer next to the endodermis. It gives rise to the lateral roots. Next to the pericycle is the vascular tissue, and we said it contains xylem and phloem. In the dicotyledonous root, the xylem forms a star shape in the center, with the phloem in between the arms. Therefore, no pith. In monocotyledonous root, the xylem alternates with phloem, forming a pith or a space at the center. Then we move to the root hairs, and we said they are microscopic outgrowth of epidermal cells, as we had discussed earlier. And we said they are numerous, long, thin-walled. Reason is to provide large surface area for water and mineral salt absorption. We move to the stem, and this is part of the plant above the ground onto which branches and leaves are attached. The main functions of the stem are support and exposure of leaves and flowers to the environment, conducting water and mineral salts, translocation of manufactured food from the leaves to other parts of the plant, storage of food as in the case of cactus, gaseous exchange in woody plants, so this is a monocot stem. The vascular bundle is distributed throughout the this space cortex. That means there is no pith, and you can see the xylem is always at the front, and then behind it is the phloem. So this is the vascular bundle. For a dicot stem, you can see it has a pith, and the phloem and the xylem are arranged in around a region called the cambium. The epidermis forms a single layer of cells enclosing other tissues. They have no chloroplast. The outer walls of the cells have waxy cuticle that prevent excessive water loss. The cuticle also protects the inner tissues from infection and mechanical injury. Next is cortex, and it is next to the epidermis. It has these tissues, cholenchyma, parenchyma, and sclerenchyma. The cholenchyma has thickened walls at the corners, and these walls are thickened by cellulose and pectin deposits, which strengthen the stem. Parenchyma, on the other hand, contains cells which are irregular in shape, thin-walled, and loosely arranged, hence creating an intercellular spaces filled with air. They are sites for packaging tissues and food storage. Some contain chloroplast and therefore able to photosynthesize. Those stems those parenchyma cells containing chloroplast and photosynthesize are called chlorenchyma. Next is the sclerenchyma tissue, and the cells are closely connected to vascular bundle, thickened by deposition of lignin through the process of lignification. Its purpose is strengthening. The pith is the central region in dicotyledonous stem, having parenchyma cells. It stores water and food substances. Some stems have no pith, hence exist as a hollow space. In monocotyledonous stems, the vascular bundle are scattered all over the stem with the phloem facing outside as the xylem faces the center of the stem. So this is the monocot. The vascular tissue is just distributed everywhere. You have the xylem facing the center, and behind the xylem is the phloem. In dicotyledonous stems, vascular bundles are arranged in a ring-like pattern around the pith. So this is the pith, and where we have the xylem and the phloem alternating. Xylem conducts water and mineral salts, as we have said. Phloem translocates dissolved food substances like sugars. We are done with that lesson. Here is an assignment for you. Just complete. All will be well. Goodbye.